Hello everyone, welcome to the student presentation week 2025-26. Myself Mr. Omkar Anil Pawar, student of B.Pharm 5th Sem of P.R. Patil Institute of Pharmacy Talegaon district Varda. Today we are going to discuss on the topic of pharmacognostic study of bitter almond. So the contents are synonyms, biological sources, geographical sources, cultivation and collection, macroscopic characters, microscopic characters, chemical constituents, solubility, extraction and isolation, chemical taste, uses, and marketed products. Now the synonyms of bitter almond: amygdala mara, kernel, nut, desert almond, almond, tropical almond, amygdalus communis, prunus dulcis, bitter apricot. Now the biological source of bitter almond. Bitter almond is obtained from the dried ripe seeds of Prunus amygdalus variety Amara, family Rosaceae. Parts use seeds of kernels. Key feature contain amygdalin, which on hydrolysis yields benzaldehyde glucose and hydrocyanic acid HCl. Geographical source of bitter almond. Bitter almond is mainly cultivated in Mediterranean regions. Important producing countries are Iran, Afghanistan, Pakistan, Syria, Turkey, Southern Europe, Spain, Italy, Greece. Also grown in California, USA on smaller scale. Cultivation of bitter almond. Bitter almond cultivation is a specialized agriculture practice distinct from the more common sweet almond. While both are from the same species, bitter almond are grown for specific industrial and culinary uses due to the presence of chemical compound called amygdalin, which gives them a bitter taste and can produce cyanide when processed incorrectly. Climate requires a Mediterranean climate, warm dry summers and mild wet winters. Soil grows best in light, well-drained loamy or sandy soils, slightly alkaline. Propagation usually propagated by seeds budding or grafting onto hardy rock stalks. Planting trees are planted at regular spacing in orchards. Flowering blossoms appear in early spring before the leaves. Collection of bitter almond. The collection of bitter almond is a crucial phase in their cultivation as it directly precedes the processing needed to make them safe for use. While the methods are similar to those used for sweet almonds, the endo-use and safety precautions are vastly different. Fruiting season. Fruits ripen in late summer to early autumn. Harvesting. Ripe fruits are hand-picked or shaken from the tree. Processing. The outer pericarp fruit wall is removed, kernels are separated from the shell, and seeds are then dried to reduce moisture content. Now the macroscopic characters of bitter almond. Bitter almond has a shape, generally oval to lanceolate, slightly compressed with one side more convex than the other. Size about 2 to 3.5 cm long and 1 to 1.5 cm broad. Surface brownish rough with fine longitudinal striations and sometimes with adhering fragments of testa. Testa seed coat. Thin, brittle, reddish brown, easily removable. Kernel whitish oily with two large thin cotyledon enclosed in a thin brown seed coat. Taste. Characteristics initially blunt but later develops a strong bitter taste due to amygdalin. Odor faint but on crushing moisturizing, it gives a characteristic odor of benzaldehyde and hydrocyanic acid. Now the microscopic characteristics of bitter almond. Testa seed coat. Outermost layer is epidermis of elongated, palisade-like cells forming a reddish-brown hard protective coat. Beneath it is a parenchymatous layer with brown pigment. Tagman, inner seed coat, formed of a thin layer of compressed cells, endosperm, usually absent or very thin, since the seed is exalbuminous. Cotyledons, made up of thin-walled parenchymatous cells. Cells are filled with numerous fixed oil globules. Also contain alluron grains, single or compound, often associated with microcrystals of calcium oxalate. Oil content. Cotyledonary cells are rich in fixed oil, fatty oil, which is the chief storage product. Chemical constituent of bitter almond. Bitter almond contains a number of chemical constituents, some of which are toxic. Here's a breakdown of the main compounds found in bitter almond. Constituents: amygdalin, cyanogenic glycoside. Its function: breakdowns to produce toxic hydrogen cyanides. Hydrogen cyanide HCl. Toxic compound. Interface with cellular respiration. Highly poisonous. Benzaldehyde. Aromatic aldehyde. Gives characteristics bitter almond smells and taste. Oleic acid. Monosaturated fatty acid. Nutritional component of almond oil. Linoleic acid. Polyunsaturated fatty acid. Essential fatty acid. Palmitic acid. Saturated fatty acid. Common in plant oils. Proteins. Nutrient globulins. Basic nutritional element. Carbohydrates. Sugar fibers. Provide energy and fiber. Vitamin E. Fat soluble antioxidant. Supports cells protection. Minerals. Magnesium, calcium, phosphate, etc. Various metabolic and structural functions. Here is the chemical structure of the bitter almond. Now the extraction and isolation of bitter almond. Extracting bitter almond primarily involves a hydrolysis step to break down the compound amygdalin into benzaldehyde and toxic hydrogen cyanide, followed by the separation and isolation of desired essential oil. The hydrogen cyanide must be safely removed to produce a non-toxic, usable product. Extraction of bitter almond oil. Preparation. The bitter almond kernels are first crushed to facilitate the release of their compounds. Hydrolysis. The crushed almonds are macerated in warm water for 12 to 24 hours. During this period, the almond's natural enzyme emulsion reacts with the amygdalin. This reaction hydrolyzes the amygdalin into glucose, benzaldehyde, and hydrogen cyanide. Steam distillation. The resulting mixture is subjected to steam distillation. This process heats the mixture, causing volatile essential oil, which contains benzaldehyde, to vaporize. The steam and oil vapor are then condensed back into liquid form. Uses of bitter almond. Bitter almond oil act as a flavoring agent, is used in bakery, confectionery and liqueurs. Pharmaceutical use. Used as a flavoring in cough syrups, emulsions and other medicines. Source of benzaldehyde. Industrially used in perfumes and cosmetics. Traditional use. Sometimes used in small amount as a sedative, antispasmodic, or for skin conditions, not common now due to toxicity. Now the solubility of bitter almond. Bitter almond oil is insoluble in water but soluble in ether and alcohol and can be dispersed in water at temperature below 15 degrees Celsius. Bitter almond protein, however, exhibits solubility similar to other plants' protein, with its solubility significantly affected by pH, salt, concentration and temperature. Solubility of bitter almond oil in water. Insoluble in water, thought it can be dispersed in water below 15 degrees Celsius. Organic solvents. Readily soluble in organic solvents such as ether and alcohol. Other solvents. Soluble in solution of nitric acid at ordinary temperature. Now the marketed preparation of bitter almond. Bitter almond oil, available as a volatile oil after removal of hydrocyanic acid, used in flavoring, perfumes and cosmetics. Benzaldehyde, isolated from a bitter almond oil, marketed as a flavoring and fragrance ingredient. Cough syrups, expectorants. Some formulations use bitter almond oil as a flavoring agent. Cosmetic creams and lotions. Bitter almond oil derivatives are used in skin care and hair products. Liqueurs and confectionery flavorings. Example, marzipan essence, amaretto flavor. From bitter almond oil, HCN free. Acknowledgement. I express my heartfelt gratitude to Dr. Nilesh Banarse sir, our estimated guide, for his continuous support, motivation and valuable guidance. I am thankful to respected principal sir, director sirs, all the faculty members and non-teaching staffs for their encouragement and assistance. Special thanks to my friends and classmates for their cooperation and moral support. Thank you for your support, guys.